Hello, welcome to the concise introduction into the science behind the stem cell treatment for lung disease using small mobile stem cells as envisioned and practiced in the future by SMS Biotech. I'm Abdulkader Ramo, and I am the Chief Scientific Officer of SMS Biotech. I would like to take you through the forthcoming slides. So the therapy is based on a single agent, which are the SMS cells that multi-target different cells. And this is for the aim of patients with lung regeneration getting their tissue regenerated. So the aim is to direct lung regeneration into individuals that needs it badly, such as in the case of COPD. Chronic obstructive pulmonary disease, COPD, is a progressive heterogeneous disease. It includes emphysema, which goes by the destruction of alveoli, and chronic bronchitis. Those two are the central pillar of this deadly disease. That disease is caused not only by smoking, but also by air pollution, by dust, fumes and chemicals and other factors, in some cases also genetic factors. They typically attack after 40 years of age and have a significant effect on airways, and air sacs. They lose their elasticity, air sacs being destroyed, the airways being thick and inflamed, and a lot of mucus is present, and these airways become clogged. So this disease can be treated to lower the symptoms, but it cannot be cured, and the damage to the lung cannot be reversed. It actually affects 15 million US citizens, causing the deaths of 140,000 per year. It's a huge economic burden, and it affects also worldwide 400 million people. The main effect of this disease is, of course, the chronic lack of oxygen. The air sacs are central to this chronic lack of oxygen. These are called the alveoli, and they are quite abundant in the lung. They represent about 480 million in the lung, and they occupy a large portion of the surface of the lung. They have a very thin wall, and they are composed of very important cells. Those are the alveolar type 1 and alveolar type 2 cells. The alveolar type 1 occupy 95% of the surface of the alveoli, and alveolar type 2 cells produce very important surfactants and are able to proliferate and generate alveolar type 1. There are other cells that are extremely important too. These are endothelial cells that provide for the capillaries and the blood supply. And of course, the extracellular matrix that occupies the wall of the alveoli and maintains its structures. Other cells are also important and are certainly occupying an important role in the lung too. We are tackling this problem of alveoli degeneration and destruction through a unique cell that has been discovered by SMSbioetech, which is the small mobile stem cells. These cells are quite fast, they are transparent, and they escape detection for a very long time. Their size is about 4 to 5 micrometer, and they have very unique physical characteristics that distinguish them from all other cells. These cells are accessible from regular blood. They can be also isolated from umbilical cord blood, but we isolate them from peripheral regular blood of adult humans. And they can be done so from individuals of different ages, and they are distinctive in appearance. Their phenotype is quite distinctive. Their speed is distinctive. They are translucent, stable, and quite resilient. They can be readily cultured and scaled up, so we can provide huge amounts of these cells using our patented technology that is being used currently to produce a lot of these cells. These cells are highly responsive to inductive medium conditions so that we can control the cell behavior. We have years of experience in doing so. We can provoke these cells to produce extracellular matrix. We can control their movement and many aspects of these important cells. They have a very important feature that is relevant to tissue regeneration, which is their selective interaction with cells that are key to tissue repair and regeneration. So they have extreme strong interactions with key cells, such as the mesenchymal stem cells that are known cells to regenerate tissue. And we have also very strong interaction with fibroblast cells in vitro, and they tend to inhibit these cells. We have also a strong interaction, in this case, that is very pertinent to lung, with alveolar type 2 cells. Now, that interaction results in proliferation and stimulation and changes in the gene expression. Now, small mobile stem cells can be cultured for many passages, meaning producing a lot of these cells, but they can also be differentiated, that means changed in other forms of cells, such as nerve cells, bone cells, and fat cells. It takes time for that to happen, but that really does not matter much with regard to the therapy that we are working at, because we rely on interaction of these cells and not their differentiation and change into other cell types. So we know also that SMS cells are easily self-organized and they are able to build a three-dimensional structure. So these cells have a tendency in vitro to form complex organized structure, which is really a good hint for them being involved in regenerative reconstruction of the tissue and organs. One important aspect of these cells is their ability to produce extracellular matrix, to secrete those proteins that are critical for structural and biochemical support of other cells. So we know that these SMS cells produce proteins that are already known to be important for tissue regeneration, such as, for example, vitronectin and heat shock proteins. We know also that extracellular matrix derived from these cells promotes the formation of small vessels from endothelial cells. And we can demonstrate that in vitro. We can demonstrate also the formation of large vessels, also through the extracellular matrix produced by SMS cells. Those large vessels that can be visible through the eye and through the naked eye. You can see here a video of the formation of these blood vessels from human endothelial cells stimulated by ECM. This is unique actually to our technology and to the capabilities that SMS cells present. The vessels can be isolated and studied further. And we know that the formation of these vessels is central to any tissue regeneration. Without the blood supply restored to a tissue, the tissue cannot grow. So the SMS cells compared to other adult stem cells or embryonic stem cells or iPSC, which are genetically modified cells, appear to be quite distinctive here. They represent safety and efficacy indeed. So SMS cells are better than embryonic stem cells and induced pluripotent stem cells because of safety. And SMS cells are having no ethical concerns whatsoever because they are derived from adult blood of adult humans and not from fetal or embryonic origin. They also are having continuous potency despite a lot of proliferation, something which is not achieved with other adult stem cells. They have also resilience, and the small size that allows them to be administered using a very particular item, which is the nebulizer. And we will see how this type of administration is unique to SMS cells. SMS cells attach to mesenchymal stem cells. Now, we know that mesenchymal stem cells are very important in tissue regeneration. We know how they modulate the immune system and lower inflammation and start the regeneration of the tissues. So having the SMS cells bind to mesenchymal stem cells and stimulate their growth and even change their gene expression appears to be extremely important, because this way we can achieve endogenous stimulation of mesenchymal stem cells by introducing the SMS cells to the body. This is an experiment where we can demonstrate how strong that binding to MSCs is. We were able to detach the MSCs from the plates where they are attached and maintain the strong binding with SMS cells, indicating that this binding between the two cells remains there for quite a bit and signifies an important biologic aspect of SMS cells. SMS cells also bind strongly to type 2 cells, not to type 1, but to type 2 cells, and stimulate their growth in the absence of serum. And that's quite important because alveolar type 2 cells, who are known to restore the lung tissue, are usually in damaged tissue, deprived of blood flow. So being able to stimulate those cells in the absence of serum is a great initiator for tissue regeneration using the SMS cells. Now, the SMS cells also change the gene expression of alveolar type 2 cells, and the changes in alveolar type 2 cells gene expression is really consistent with the healing of the organ, because the changes in those expressions, once studied in detail, they indicate that SMS cells causes a lowering of inflammation, reduces cell death like apoptosis, and enhances pathogen protection, promotes the formation of blood vessels, angiogenesis, reduces vessel permeability, and lowers blood pressure, something which is often accompanying COPD, which is hypertension. So we observe also an important aspect related to tissue regeneration, which is the suppression of tissue fibrosis. If you get a repair of the tissue that is based on fibrotic cells, you don't get an ideal tissue. Actually, you may get the tissue damaged, permanently damaged through that. So suppressing the tissue fibrosis and enhancing cell parenchyma really is what brings the tissue back. So that is what we observe through the changes in gene expression. We observe downregulation of the genes that are responsible for tissue fibrosis, and the upregulation, the stimulation of tissue that inhibit, of genes that inhibit the tissue fibrosis. So in addition to all that, the cells, the size of the cells, the small size of these SMS cells, in addition to their resilience, made it possible for us to generate a mist using a nebulizer that can be inhaled. In fact, we can apply these cells through a nebulizer, create that mist, and maintain the full functions of the cells. Something that is unique to these cells because of their unique size and unique resilience, something that cannot be achieved with any other cell. So we administer the cells directly to the lung and maintain the locality of the effect. So we don't lose cells to other organs. We can concentrate the effect on the lung organ. We tested using animals the safety of these cells. We used intravenous injections. We used direct application through the trachea to the lung. And we use a large number of these cells, and we observed no adverse symptoms, no lesions, no temperature changes, normal appearance, normal behavior, and there was no adverse effect whatsoever. In fact, we also tested whether the cells produce any tumor. This is a test that the FDA requires, and we injected the cells not just in one place, but in many tissues. We injected them subcutaneously, intramuscular, intratibial, and in the lung cavity. No tumors were observed whatsoever. The cells are adult stem cells. They are safe. But what is most important is that we observe that when these cells are injected to destroyed lungs, to lungs that have destroyed alveoli, a typical model for emphysema, a standard animal model is the one that has these alveoli destroyed. When these cells were administered to the lung after the destruction of the alveoli, we observe quick regeneration. So in fact, in 1 week, we can observe almost 100% regeneration of the alveoli, and the animals are able to perform as well as the controls in terms of velocity and endurance through an exercise test. That is done through a spreadsheet. So the cell therapy, the upcoming cell therapy for emphysema COPD will be based on the resilience of these cells, their small size, where we can administer these cells using a nebulizer, and we will stimulate alveolar type 2 proliferation, and causing, therefore, the production and regeneration of type 1, and therefore restore alveolar cells. We will modify the activity of many genes related to alveolar type 2 that are critical to lung repair and regeneration. We will stimulate also mesenchymal stem cell proliferation and reduce inflammation and support tissue regeneration. We will stimulate a capillary formation and provide access to blood for the damaged tissue. These are the main features and the cornerstones of this unique cell therapy. This is an extraordinary opportunity to invest in a cell therapy for lung treatment, and SMS cell therapy prospects for lung regeneration are really exceptional and not to be non-comparable to any other available so far. So SMS cell will target several types of cell key to tissue regeneration, as has been shown in the bench many times. SMS cells stimulate the proliferation of alveolar type 2 cells that are key to lung regeneration. This is a given in the current research and understanding about the tissue regeneration. SMS cells activate mesenchymal stem cells that are endogenous promoters of tissue repair in the lung. SMS cells will inhibit fibroblast cell proliferation, the main cause of lung fibrosis. And unlike mesenchymal stem cells, SMS cells are able to proliferate extensively without loss in potency. SMS cells are sturdier, more robust, easier to handle and administer. Their robustness allows, for example, the shipment at refrigerator temperature all over the world. SMS cells can be administered using nebulizer and introduced to this lung directly, as we have seen. And SMS cells are unlikely to induce any significant immune reaction because of the absence of molecular targets for immune rejection. And SMS cells are adult stem cells and are the safer alternative to embryonic and genetically engineered cells. The bloodstream is one of these cells' natural habitat, and that is unlike mesenchymal stem cells. SMS cells demonstrate in vivo using animal model an extraordinary safety and efficacy profile. So we have multiple manufacturing advantages coming with these cells. And because of that, we're going to end up providing an allogenic, off-the-shelf and scalable production. This is a production that is patented, it's proprietary. It's a production that is economic, meaning that we can provide this therapy at reasonable pricing, centrally manufactured and shipped all over the countries and also worldwide. So this is shipping at a storage temperature of 4 degrees, something also unique to these cells. SMS cells provide availability, stability, binding, effect at molecular and organ level, and safety, good safety profile. They demonstrated through preclinical studies and soon, we hope, through clinical studies. So these human small mobile stem cells are a next generation stem cell regenerative therapy. There is no genetic manipulation required. It's a direct route to the lung. And it's a multi-targeted, orchestrated, smart, intelligent process, natural process, off-the-shelf cell therapy, targeting chronic widespread indications and aiming for cure, not mere treatment. Thank you very much for your attention, and please go back to me for any questions.